Without further ado, may I invite the first group to join the stage with such an interesting topic about feasibility of generation and expansion of hematopoietic stem cell derived natural killer cells for a potential alternative cell source of cancer immunotherapy, presented by Poonnattha Anantasaeree from Faculty of Medicine, Prince of Songkla University.
Good morning, everyone. My name is Poonnattha Anantasaeree from Prince of Songkla University. Today, I'm going to present my research project entitled Optimizing the Differentiation and Expansion Protocols of Mobilized Hematopoietic Stem Cells Derived Natural Killer Cells for Cancer Immunotherapy. Hematologic cancers are cancers of the blood and bone marrow. This disease arises from abnormal hematopoietic or lymphoid cells that grow uncontrollably. Despite the availability of chemotherapy, radiotherapy, targeted therapy, and stem cells transplantation, many patients still relapse and treatment-related toxicity. This highlights the need for safer and more effective immune-based therapies. As a result, we're particularly interested in developing immunotherapy. Among these approaches, adoptive cell therapy or cellular therapy is especially promising. In adoptive cell therapy, immune cells are collected from the patient or donor, and after that, activated and modified outside the body and then fused back to fight the cancer inside the body. The best known example is CAR T therapy, which has shown remarkable clinical success, but also come with major limitations. Because of natural biology of T cell, CAR T cell requires specific recognition to be activated. Moreover, it can cause life-threatening complications such as cytokine releasing syndrome and neurotoxicity. This drawback highlight the need for safer, scalable, and more universally applicable cell-based therapy. Unlike T cell, NK cell function at the interface between innate and adaptive immune system and do not require prior antigen sensitization to exert cytotoxicity. Importantly, NK cells do not induce graft versus host disease. However, NK therapy still faces key challenges: inducing donor-to-donor viability, limitation expansion potential, and functional exhaustion. Different cellular sources offer different advantages and limitations. Among these, hematopoietic stem cells show self-renewable and differentiated capacity, making them highly attractive for scalable NK cells generation. The purpose of our study, the first one is to optimize the differentiation and expansion protocols of HSCNK or hematopoietic stem cells derived natural killer cells. And the second one is to compare their phenotypes and functional activities with those of peripheral blood derived natural killer cells or PBNK. Now that we understood the goals of this study, let's move on to how we actually conduct the experiment. The overall workflow study can be divided into 4 main phases: cell isolation, cell culture, NK cell expansion, and functional assay. I will walk through each step along with the corresponding result in the upcoming slides. We began by isolating 2 cell populations from mobilized hematopoietic peripheral blood: CD34, which is representative of hematopoietic stem cells, and CD56 NK cells, using magnetic separation. This procedure yields 92% purity for stem cells and 78% purity for NK cells. Next, in cell culture phase, stem cells were generated into 2 phases culture system. The first process supports lymphoid progenitor expansion, and the second phase drove NK cell differentiate over 28 days. During this stage, PBNK are cryopreserved in liquid nitrogen and later thawed for expansion step. For the expansion phase, both HSCNK and PBNK were co-cultured with Epstein-Barr virus lymphoblastoid cell line or EBV-LC feeder cells. The HSCNK cells achieved a cumulative around 294 expansion. After expansion, we assessed their phenotypes, and both populations expressed CD56, a hallmark of NK identity, and CD16 typically low in naive NK cells and higher in activated NK cells. Follow the expect pattern. HSC-NK cells show comparable activating receptor to PBNK and also demonstrate strong expression of NKp44 indicating enhanced activation potential. They also exhibit markedly reduced inhibitory receptor expression, further supporting and activating phenotypes. Additionally, CD62L was more prominent in HSC-NK cells, suggesting the improved capacity for tumor cells engaged in apoptotic induction. Finally, we expand NK cells underwent functional assays, including proliferation, degranulation, and intracellular cytokine secretion to confirm their anti-tumor activity. HSC-NK saw no significant reduce in tumor target recognition compared with HSC-NK. For specific cytolysis, we use K562 leukemia cells as the target. So HSC-NK cells demonstrate high and specific cytotoxicity activity comparable to PBNK cells. In summary, our optimized protocols enable robust expansion and functional activation of HSC-NK. These findings support their potential as reliable off-the-shelf allogenic NK production for cancer immunotherapy. Looking ahead, the next step to enhance their therapeutic potency through CAR-NK engineering combined with immune checkpoints inhibitors to further improve their tumor recognition, persistence, and anti-tumor activity. That's it for today. Thank you.
So your HSC NK cell is a monoclonal, or because you isolate the HSC NK cell for the what is called? It's
peripheral blood mononuclear cell, right?
Right.
Yeah.
And so how do you isolate it to make sure that it's a monoclonal?
We isolate from all peripheral blood to be monoclonal antibody by leukapheresis. The procedure called leukapheresis, and after that separate to NK cells and stem cells by use magnetic separation.
Can you summarize again what is the advantage that you propose to use NK cell from hematopoietic derived versus PBNK?
The PBNK cells are like the patient that come to the hospital. Before that, do some immunotherapy. They're faced with chemotherapy, so the immuno cells also be like compressed and inadequate to do in the PBNK cell. That's why we focus on HSC that have ability to self-renewable and differentiate the expansion capacity. So we focus on HSC to extend the capacity and feed it with in cytokines and the environment that mimic the tumor to activate their function.
So in terms of the specificity between CAR-NK from your work and CAR-T, so could you compare between these two cells?
Okay, so my work is not CAR-NK, it's just NK cell, but we focus on NK cells because in T cells, lots of patients facing with cytokine releasing syndrome and neurotoxicity, which is a limitation of a CAR-T cell. After patient got the CAR-T cell, they can be like high risk to be like cytokine releasing syndrome. But in CAR-NK or in NK cells, naturally, NK cells do not cause of neurotoxicity because of the mechanism of immune response. T cell is adaptive immunity, which require specific recognition to exert the cytotoxicity, while NK cells it interface between innate and adaptive, and its response is high ability while cause lower damage than T cell to the health patient. Thank you.